All right, so we are recording. We can start. So today we are going to look at nucleic acid chemistry, and it will be a very interesting topic because we'll be basically looking at the nucleic acids, what makes them up. But of interest is a group of drugs called the nucleoside reverse transcriptase inhibitors. So by the end of this class, you should be able to understand how this particular class of drugs works, or they're antiretroviral drugs and they're used in the treatment of HIV and AIDS. And actually, there's one drug called zidovudine, which was the first drug to be used in HIV-1. So zidovudine is a nucleoside reverse transcriptase inhibitor. What that means is um, any drug that is called an analog of something, it just means that it's a compound that looks alike with a particular compound. In this case, zidovudine looks like it's a nucleoside. Okay, so since it looks like a nucleoside, an enzyme called reverse transcriptase binds to the drug instead of binding to the actual substrate, which is just normal nucleosides. Okay, so if we understand what a nucleoside is, we'll be able to understand how zidovudine works. In this case, the the the, the enzyme called reverse transcriptase binds to the drug instead of binding to the actual substrate that it normally binds to. In the process, the drug causes a block of the enzyme. This is like suicide inhibition. Okay, soon we're going to learn about enzyme classification and catalysis. So you guys are going to see um, uh, the different modes through which enzymes can act. Okay, people who can't relate in class, they do inconvenience so much. Right. So we're talking about the dopamine. The dopamine is a drug that is used in treatment of HIV and AIDS. It's actually mostly what it is used drug, which is the oldest. Technically, we call it azithromycin. So azithromycin or zidovudine is a nucleoside reverse transcriptase inhibitor. So it blocks an enzyme called reverse transcriptase. So this enzyme is found in HIV, in the HIV virus, and it is the major culprit in this disease. So it's a major culprit in this viral infection. So reverse transcriptase is blocked through a group of drugs called nucleoside reverse transcriptase inhibitors. Right. So you should know that the nucleotides and DNA and RNA. So synthesis of DNA and RNA comes from the nucleotides. So we need to know what a nucleotide is made up of. People can't relate in classes. It's so inconvenient. Okay, so a nucleotide is a precursor for the synthesis of DNA and RNA, okay, in a cell. Then you should also remember that the nucleic acids, their their major function is for storage and transfer of genetic information. So we know that DNA is there for storage of genetic information, and we know that RNA is there to pass on that information from DNA all the way to protein synthesis. Okay, so you should also remember that the universal energy currency, which is ATP, sorry, adenosine triphosphate, is actually a nucleotide. Okay, so we need to know what a nucleotide is. It's a precursor for the synthesis of DNA and RNA. We also learned that ATP, which is the major energy currency, is actually a nucleotide, and um, we have many coenzymes like organic compounds that work along with an enzyme to make it function properly. Okay, so in this case we have oxidized nicotinamide adenine dinucleotide. So NAD plus. We have FAD, which is flavin adenine dinucleotide. You need to remember these molecules because when we start doing metabolic chemistry, you are going to find them in pathways such as the glycolytic pathway, the tricarboxylic acid cycle, the pentose phosphate pathway, and other pathways through which they act as coenzymes. Okay, so we shouldn't worry about these compounds because we are going to meet them again. We need to remember what they are. They are actually nucleotides. Okay, so um, people should remember things like cyclic AMP, which is cyclic adenosine monophosphate. We should remember that we also have cyclic guanosine monophosphate, which is a compound that is used that the body uses to relax the smooth muscles. So smooth muscle relaxation is possible because of a molecule called cyclic GMP. Okay, so cyclic GMP is a second messenger molecule that is used to cause smooth muscle relaxation. Okay, we have drugs such as um, sildenafil nitrate. So sildenafil nitrate is a drug that is used in erectile dysfunction. Okay, so in erectile dysfunction, it appears that there is massive degradation of cyclic GMP to non-viable products. So in this category of men, so this disease is only for a man. In this um, in this disease, the cyclic GMP is converted immediately. It is converted to non-viable products that are unable to cause smooth muscle relaxation. So when we give a drug called sildenafil, it blocks an
Can
okay
you
type
repeat?
5.
You are
So
like
this
skipping.
enzyme is responsible. Oh, I'm skipping.
Yes, you are skipping.
You
Then
win.
Am I skipping everywhere to everyone?
No, now you are.
Yes, sir, you were skipping at some point.
Oh, I see.
Yes, you're skipping.
Am I still skipping?
No.
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 Yes.
All right. So I was talking about cyclic GMP. So cyclic GMP as it's a nucleotide, right? So cyclic GMP um it's actually used by the human body. Okay, it is used by the human body to relax the smooth muscles. When the smooth muscles are relaxed, what happens is that the blood will actually be flowing in a normal way. Okay, I gave an example of erectile dysfunction. A man with erectile dysfunction. A man with erectile dysfunction, their penis is unable to erect for a long period of time. Okay, why? Because their blood vessels, they are stiff. They cannot allow blood to pass through. Okay, and their veins, they actually drains the blood real quick. So now there's a drug called sildenafil nitrate. Okay, so sildenafil nitrate is actually blue diamond. It's called blue diamond amongst the young men because they abuse it. So blue diamond or sildenafil nitrate is a drug that blocks an enzyme called phosphodiesterase type 5. Now, what's the function of phosphodiesterase type 5? This enzyme is responsible for the conversion of cyclic GMP or catabolism of cyclic GMP to other non-viable products that are unable to cause smooth muscle relaxation. In this disease, the person is metabolizing the cyclic GMP real quick, like very fast. So there's no time for the cyclic GMP to cause smooth muscle relaxation and allow blood flow to go to the penis for an erection to occur. Because for an erection to occur, you need a lot of blood to go to the penis. So for that to happen, you need the blood vessels to be relaxed, and then they will allow as much blood as possible. Now, as we have already seen that in these men, the blood is unable to pass. Why? Because the blood vessels are stiff. There's no smooth muscle relaxation. So this drug comes and blocks the enzyme called phosphodiesterase type 5. So PDE type 5 is an enzyme that is blocked in sildenafil nitrate um, therapy. Okay, so you should remember cyclic GMP because even in the next academic year, when you come to do pharmacology and therapeutics, I'll mention the same thing again. So in this course, you just remember enzymes and intermediates and pathways. Okay, for now we are doing structures, so you need to practice how to draw these structures. Okay, so compounds like FAD, which is flavin adenine dinucleotide, NAD plus, which is oxidized nicotinamide adenine dinucleotide, and other metabolic regulators such as cyclic AMP and cyclic GMP. Okay, they are nucleotides. Okay, so that was an introduction. Today's class, by the end of this class, students should be able to understand and explain the following. You should be able to draw structures and and the properties of the purine and the pyrimidine bases. Okay, so in Bio 1400 we learned about purines and the pyrimidine and nitrogenous bases. So in this course, students should be able to draw the structures on their own. Why? Because this is structural biochemistry. We want you to learn how to draw the structures. Why are we asking you to draw the structures? Because at a later stage, when we come back to doing nucleic acid metabolism, we'll see how the liver or the hepatocytes synthesizes the purines and the pyrimidine nitrogenous bases. So it, it is very wise for us to know how to draw these structures. Okay. So by the end of this class, you should be able to draw the structures of the purines and the pyrimidines. Okay. It's not like 1400 where they used to give you just MCQs alone. No. Okay. Then you should also give us the properties of the purines and the pyrimidines. By the end of this class, students should be able to tell us what a nucleoside is, should be able to tell us what a nucleotide is, and of course, you need to tell us what nucleic acids are, and the structures, of course. We are still doing structural biochem. Remember that. So we haven't graduated from this. By the end of this class, you should be able to tell us the use of the nucleoside analogues. An analog is a compound that looks similar with a non-compound. In this case, we know what nucleotides are. So we have nucleosides, analogues. These are drugs that are used in HIV. That's why we're talking about HIV biochemistry. So the use of nucleoside analogues as therapeutics. Therapeutics means applying the pharmacology in the treatment of diseases. Okay. We should also have an overview of cancer, how cancer comes about, the mutations that happens on the level of the nuclear, the nitrogenous bases, and also HIV biochemistry. So HIV biochemistry will basically talk about the enzyme, reverse transcriptase, and other enzymes, DNA polymerase, and inhibitors. Okay, because we also talk about other diseases like mollidoqualesa. If you have heard of mollidoqualesa, we call it a herpes zoster, VZV, varicella zoster virus. Okay, so we also talk about a drug called acyclovir, which is used in the treatment of mollidoqualesa. We should also
Sir,
just
you
read
keep
about genetic
skipping.
mutations. Oh, I keep skipping.
Like very much now this time. I can't even get you.
It has become worse. Okay. I'm not going to log out. I'll just connect with different network so that let me just connect to a different network. Am I still skipping?
No.
Okay, where did people stop getting me?
We're talking about what we'll know after like we finish learning.
Oh, okay. Did we reach the end?
So we reached the last
Yeah.
point.
Okay, okay. So we're saying that we also have other drugs that are used to treat other viral infections like varicella zoster viral infection, which is traditionally called muliro wa kwalesaiye in Zambia. Okay, so there's a drug called acyclovir. It's a nucleoside analog, and we're going to see how these drugs works as nucleoside analogs. Can people hear me?
Yes.
Okay. All right. So that is what we are going to learn. We're going to learn about the structures of the purines and the pyridines, the nucleosides, nucleotide, and nucleic acids, and the application of nucleoside analogs in HIV medicine. Right. So we start with a nucleotide. Can someone tell us what a nucleotide is? Anyone tell us what a nucleotide is? Sadia, what's a nucleotide? Hello?
A nucleotide a molecule composed of a nitrogenous base, a pentose sugar, and a phosphate.
Right. So a nucleotide is made up of three components. That's a nitrogenous base attached to a sugar molecule, which is a pentose sugar, and to the pentose sugar, there is a phosphate group or phosphate groups attached to it, which makes a nucleotide. What of a nucleoside?
It's a nucleotide without a phosphate group.
So a nucleotide without a phosphate group is called a nucleoside. So we have these drugs are not nucleotide reverse transcriptase inhibitors. No, these drugs are nucleoside reverse transcriptase inhibitors. Right. So we know what a nucleotide is. We know what a nucleoside is. Now let's see this. We mentioned that a nucleotide should have three components: a nitrogenous base, a pentose sugar, then the phosphate group. So the nitrogenous base can either be a purine or a pyrimidine. Okay, let me assure you, the body actually takes so much time for it to synthesize these purines, especially the purines. Okay, so we have about 15 steps that the liver cells uses for them to synthesize a purine. Okay, anyone to give us an example of a purine base?
Guanine.
Guanine, yes. What else? Pauline, what's the other um, purine base? Florence, people are writing messages. Me, I can't see. Me, I can't see.
Uracil.
Sorry,
Uracil, sir.
is uracil a purine?
Adenine.
Adenine. So adenine, yes. Uracil is not a purine. Uracil is a what? Sadia, uracil is a what?
A pyrimidine.
Uracil is a pyrimidine. Okay. So the other nitrogenous bases that we have are called pyrimidines. Okay. The pyrimidines, we have uracil, we have thiamine, and Eliza. What's the other pyrimidine?
We have cytosine.
Cytosine, yes. So now, what's the difference between a purine and a pyrimidine? Florence, what's the difference between a purine and a pyrimidine? Mukata, what's the difference between a purine and a pyrimidine? Emmanuel, if you're in class, what's the difference between a purine and a pyrimidine?
Can I answer, sir?
Yes, please. What's your name?
It's Mukata.
Okay, Mukata, yes, go on.
The difference between the purine and the pyrimidine
Yes.
that the other one just let me be specific. Purine has three nitrogenous bases, and then pyrimidine has just two. Yes, that's one difference. And then the purine, the structure, it has wrong structure compared to pyrimidine.
Repeat
Maybe
the
others
first
can add on.
repeat the first
The,
difference.
the first difference is that uh, purine, it has three cytos. It has three nitrogenous bases, and then pyrimidine has just two.
No, that's
Is it
politics
clear?
now. One day you can unmute yourself. That's politics.
Uh,
I can't hear you.
hmm.
I didn't get anything. Is anyone able to hear her?
No.
No one is is able to get her. She's even left.
Maybe she's quiet.
No, she's not quiet. She's left. Eunice, what's the difference between a purine and a pyrimidine? Eunice, unmute yourself, or I'm going to unmute you if you don't unmute yourself. I'll unmute you by force so that you can speak. Eunice, what's the difference between a purine and a pyrimidine? People can't hear me again? Or she's not just responding?
Uh, the other one just doing,
Which
and the
one is
other
that?
one just
Emmanuel,
doing.
you have also started politics. Um, Mike, what's the difference between a purine and a pyrimidine? One day we couldn't hear you. If you can speak up.
Can you hear me now?
Yes, you can.
I was saying that a purine is a two-ringed structure, while a pyrimidine is a single-ringed structure.
Yes, so a purine is a double ring compound, whereas a pyrimidine is single ring. So now, how does the cells know that I'm now synthesizing a purine or I'm now synthesizing a pyrimidine? Sadia, how does the hepatocyte knows that right now I'm synthesizing a purine, on the other end I'm synthesizing a pyrimidine? How does that happen?
I don't know, sir.
Okay, fair enough. Who wants to answer that question? Eliza, would you love to answer that question? Florence, Mike, there are some students who don't want to speak. Hope, how does the cell knows that right now I need to start synthesizing a purine? And you know the steps for purine biosynthesis, it's actually 15 steps. Okay, step 1, this happens, step 2, this happens, step 3, this happens, step 4, up to step 15. Okay, so how does the cell know? Oh, unmute yourself and tell us how that happens. Matthews, you are busy asking questions in the groups. It's now time for you to answer the questions. Pauline, unmute yourself. Okay, so the cell knows because there are some enzymes. We have enzymes that are just specific for purine biosynthesis. We also have enzymes that are just unique for pyrimidine biosynthesis. So that's how the cell knows that right now I'm synthesizing a purine, or right now I'm synthesizing a pyrimidine. Okay, next week we'll do enzymes. So you're going to learn about the enzymes and everything else. Okay, we can go back. Okay, the other component is a pentose sugar. So we have two sugars in here, two pentose sugars. Florence, unmute yourself and tell us the two different sugars that we have that are found in the nucleotides.
A ribose sugar and a deoxyribose sugar.
a ribose sugar and a deoxyribose sugar. This is structural biochemistry. If you don't know how to draw the structures, then you are not doing anything and you are not helping yourself. Okay? And you are not helping us because we want to produce people who are very brilliant, who knows what they are doing if they have a patient. Okay? So we have a ribose and a deoxyribose. A ribose sugar is found in RNA, in DNA. True or false? One die, true or false?
False.
Why?
The ribose sugar is found in RNA, while the deoxyribose sugar is found in DNA.
Very good. So ribose, RNA, deoxyribose, DNA. Okay? We're going to see the structures soon. So don't worry about that. We're going to see the structures soon. Then the last component is it's a phosphate group. This is actually inorganic phosphate. Okay, it's inorganic phosphate. We call it phosphoryl group. So we can have one, which is mono, two, di, three, tri. So we can have a monophosphate, a diphosphate, and a triphosphate. We can't go beyond that. Okay, as you increase the phosphate groups, you're increasing the energy potential of that particular compound. That's why ATP has more energy than ADP, and ADP has more energy than AMP. Okay, so we need to understand latecomers. Wow, people coming late in class. You know that this is not allowed, especially they are going to be chasing you. So Dr. Mkape is going to be chasing people who comes late in class. This is not allowed. And if they spot you, you are likely to fail. So don't come late. Don't don't do funny things like for boys. You are going with a lot of hair. You're not going for a barber shop, guys. You are not having a tie. If they spot you, you don't have a tie. The chances of clearing a course will be slim. So just wear normal clothes. You have a tie, proper clothing. They don't want your body. There are no your bodies in medicine. There are no slay queens in medicine. That's why they do separate slay queens from actual students. They do separate your bodies from actual students. Okay? And this is the academic year where they do that. So just be a normal student. That was just an advice. Right. So the phosphate group is esterified to the sugar, which is the pentose sugar, okay, which is actually a furanose ring because it's five-sided. Okay, so esterification just means a reaction between a, a carboxylic acid, okay, an acid and an alcohol. So that's what esterification is, to form an ester. So there's an ester linkage between the phosphate group and the pentose sugar. Okay, so when the base combines with the pentose sugar, it forms a nucleoside. Okay, so a nucleoside is a combination of a base and a pentose sugar. Now, when the nucleoside undergoes esterification to to a phosphate group, it forms a different compound we call a nucleotide, okay, or a nucleoside monophosphate. So nucleoside monophosphate. Then when a second phosphate group is attached to the nucleoside monophosphate, um, it forms a nucleoside diphosphate. Then when a third phosphate group is attached, it results into the formation of a nucleoside triphosphate. Okay. Then the nucleotide, when they combine together, they will form either RNA or DNA, which are nucleic acids. So RNA and DNA are actually polymers of nucleotides, not nucleosides. Okay. And you should take note, it's nucleoside monophosphate, not diphosphate, not triphosphate. Okay, now we go into the details of the nitrogenous bases. We need to see the structures because this is structural biochemistry. So we have two types as we have already established, the pyrimidine and the purines. We'll start with the purines. So these ones are double ringed. Here we have two common ones, which are adenine and guanine. So adenine and guanine can either occur in RNA um, or DNA. What differentiates DNA and RNA is a pyrimidine base, uracil, and thiamine. Okay. So the purine that is present in the DNA and RNA are the same. That is adenine and guanine. Okay. In chemistry, adenine is a 6-amino purine, okay, and guanine is a 2-amino 6-oxypurine, okay. So the structure of adenine is that of an, an amine which has a purine which has got an amino group attached to it at position number 6. You need to know how to count, guys. You need to know how to count the numbers on the rings. Okay, they are not carbons; they are just numbers. Okay, then the guanine is a 2-amino 6-oxypurine. What that means is that at position 2, there's an amino group attached to it. At position 6, there is an oxygen attached to it. Okay, so now a purine has got a general structure. Okay, a purine has got a general structure which you are supposed to learn. When you learn the general structure of the purine, you'll be able to draw the structure of adenine and guanine because this is second year, this is not first year where you don't draw. You have to draw this time around. So now, the numbering of the purine ring with the structures of the adenine and guanine are shown in this diagram. So here you can see, let's count together. So this is the general structure of a purine base. Okay, so here you can see this nitrogen here. Okay, this nitrogen here is the first. So this is number 1. So you go anti-clockwise. Okay, you go anti-clockwise. For the pyrimidines, it's different. You're going to see it later. So this is number 1. This is not 2. This is 6. Okay, if you make a mistake, you will say this is 6 when it is not. So this is 1, 2, 3, 4, 5, 6. Okay. So now here the importance of numbering them is because you want to know where the functional group is attached. We said adenine is 6-amino purine, okay, meaning at position 6 here there's an amino group attached to it. Look at this molecule. This whole thing is just a purine nitrogenous base, but at position 6 there's an amino group attached to it. In medicine, classifying things is very important because you don't have too much memory or actually time to start keeping details like these. Okay, so here you just know that at position 6 there is an amino group attached to it. So here you can see you need to know how to draw this. So 1, 2, here there's just a carbon, here next nitrogen. Okay, so you do something that is 6 numbered. Okay, then you do double bond here, then you skip, you go to another one, double bond, you skip, you go to another one, double bond. Okay, it's very easy. It looks like a benzene ring, but it's not benzene. Why? Because we have nitrogen instead of carbon. So it's like a benzene ring. Then this side it has got it's five-sided, like 1, 2, 3, 4, 5. But it continues. Okay. So 1, 2, 3, 4, 5, 6. You end here, then you start again. 7, 8, 9. So meaning a purine has got 9 positions. Now
I can't get you.
Can people hear me?
Yes, sir.
Okay. Whoever texted me, thank you very much. I I I didn't even know that I had left the meeting because I'm not seeing anything. Where did people stop hearing me?
Teaching us how to count the rings.
Okay, how to count the rings and how to draw the structures of adenine and guanine. So this is a general structure. Once you memorize, there's a benzene ring. It looks like a benzene ring, but then it's not a benzene ring. Okay, so instead of being putting carbon here, you put nitrogen. You skip carbon, then after the carbon, nitrogen.